Hello, everyone. My name is Andrew Fitzgerald from ICF Next. I would like to welcome you to our Environmental Epidemiology webinar series. A few comments before we get started. All lines have been muted upon entry and will remain muted for the duration of the webinar. If you experience technical difficulties, please use the chat feature on the right-hand side of your screen and contact the host of the webinar to assist you. We encourage you to submit questions by using the Q&A feature. Please type in your question and select all panelists before hitting submit. We will moderate the questions on your behalf during the Q&A portion of the presentation. Feel free to submit your question at any time. Unfortunately, we may not be able to get to all questions sent in due to time constraints. If you require closed captioning, please refer to the link in the chat panel that will be posted momentarily, and you'll just be asked to enter your name. And with that, I will turn it over to Kirk.
Thank you, Andrew. Yes, hello and welcome, everyone. Thank you for taking time this afternoon to join this webinar, Key Gaps in Environmental Epidemiology and Exposomics. I'm Kurt Del Valle. I'm a program director in the Environmental Epi branch in the Division of Cancer Control and Population Sciences at NCI. And I'm also joined by my colleague, Gabriel Lai. We're thrilled to have our two speakers, Dr. Robert Wright and Jessie Buckley, here to share their research and thoughts and needs in the field in these two fields. This webinar was organized as part of an effort to promote and advance the science around environmental exposures and health at NCI and NIH more broadly. I think with the Human Genome Project, we saw tremendous advances with the way genomic research is conducted, from sequencing technology to advances in bioinformatics. That really propelled the field of genomics and the ability to investigate genetic drivers of disease. But it also revealed that for almost all diseases, genomic factors are probabilistic risk factors and not deterministic of disease or disease outcomes. And environment, and speaking of environment broadly here as everything not genetic, is as big of a piece of the disease puzzle. There is, however, an identified need to better understand the complex relationships between our physical, chemical, and social environments and disease risk and outcomes. So I'm happy that today we get to have an opportunity to continue the discussion around the opportunities to advance the field of environmental health research. So as Andrew stated, please at any time put your questions in the Q&A box, and at the end of both talks we'll have a chance to answer those questions. And now I'd like to introduce our first speaker, Dr. Robert Wright. Dr. Wright is the Ethel H. Wise Chair of the Department of Environmental Medicine and Public Health, and also the co-director of the Institute of Exposomic Research, and also the founder and director of the Frank Lautenberg Environmental Health Sciences Laboratory at Mount Sinai Icahn School of Medicine. In addition to being a pediatrician, his research focus is on chemical mixtures and exposomic approaches to investigate the effects of environmental factors on child health, particularly neurodevelopment and reproductive health, as well as evaluating the role of genomic factors in modifying chemical toxicity. He's a member of the National Advisory Environmental Health Sciences Council, and he's also currently PI for the Mount Sinai Lab Hub for the CHEER and HERE programs. So Dr. Wright, I'll turn it over to you.
Thank you, Kurt. Can you hear me?
Yep, we can hear you.
So for those of you who are literature majors, you might notice that the title of my talk, Where Have We Been and Where We're Going, is similar to a Joyce Carol's short story. And if you've read that short story, you might find that disturbing, but I'll leave it at that. So I'm going to talk about where we've been, which is the exposome. So the last 16 years, we've really been trying to define the exposome. And the term itself was first coined by Christopher Wild, who's now the director at IARC. And he referred to it as all the environmental exposures throughout a lifetime. I think that's perhaps a bit too broad, so I've sort of reduced it a little bit to all the health relevant environmental exposures throughout the lifetime. But he proposed it as being the analog to the genome, so that we would be able to actually match the genome in terms of the scope of what we measure. And the internal exposome is actually a function of the external exposome, which is something that we'll talk about a little bit more going forward. But there are things we can measure inside the body. There are things that we can only measure outside the body. But keep in mind, everything inside the body was once outside the body, including our DNA. Everything that is in our bodies was something we either ingested or breathed into our bodies. The exposome is actually much bigger than the genome, and it varies over time and space. So we often talk about how it varies over time, and sometimes we forget that it varies geographically as well. And that is something that's actually going to be very important as we go forward and try to measure and understand the exposome. So that's where we've been. So where are we going? Well, healthcare is really dominated today by complex diseases. The one exposure or one risk factor as a cause of disease, such as what we see from Mendelian diseases or infectious diseases, is actually relatively rare for the major drivers of healthcare costs. And these are diseases such as Alzheimer's disease, Parkinson's disease, heart disease, COPD, in children, autism, dyslexia, learning disabilities, and cancers and obesity. All of these are complex diseases, and all of them are caused by both genetic predisposition and environmental exposure. So if that's where we're going and precision medicine is coming with us, then we have to also define precision medicine. Precision medicine is really defined as the tailoring of medical treatment to the characteristic of each individual patient, so that we can have interventions that are concentrated on those who benefit, sparing the expense and side effects for those who do not. But precision means we have to understand each patient's background, and we have to understand how complicated that background is. It's not just genetics, but it's also the social environment, the nutritional environment, the physical and the chemical environment. So if precision medicine is going to work, it really can't be restricted to just genomic background. And we've known this for a long time. In fact, this is the cover of Science in 2002. It was a special issue on complex diseases, and it talked about how genes, behavior, nutrition, infections, chemicals, the physical environment, culture, society, and stress all play a role in complex diseases, and it viewed these diseases as puzzles. And in fact, the introduction to that issue was actually entitled, "It's Not Just the Genes." And it listed all the complex diseases that I listed before as being the major drivers of healthcare. And it presented this as a puzzle. And I would argue that this puzzle needs to include genomics and epigenomics, transcriptomics and proteomics, but it also needs to include the environment. It needs to include nutrition, social environment, chemicals, physical environment, if we're going to be able to complete this puzzle to finally understand how to do precision medicine precisely. So here's some examples. So this is actually a very commonly used example for the importance of precision medicine. And this would actually work. So say a 53-year-old woman develops deep venous thrombosis on a trans-Pacific flight. She develops a GI bleed after she starts a standard dose of warfarin. Well, the reason this happened is because she has a genetic variant that makes her a slow metabolizer of warfarin, which is a blood thinner used for deep venous thrombosis. And if they had done a genome screen in advance, they would have started her on a lower dose of warfarin because they would have understood that she was a slow metabolizer. But what about this example? A 17-year-old child with autism has a noticeable increase in head banging and anger outbursts. We think of autism as a genetic disease, but children with autism, adults with autism, live in the same environment as the rest of us, and they're exposed to the same chemicals that the rest of us are exposed to. And in fact, children and adults with autism often have pica, they eat non-food substances, and therefore there may be at greater risk for certain types of environmental exposures. This child actually turned out to be lead poisoned, and when the lead poisoning was treated, he got much better in terms of his behaviors. And it turned out that he was eating newspaper, and newspaper ink had lead in it, and it had enough newspaper ink that he was ingesting that he's getting severely lead poisoned. A genome scan would not have predicted that. And then another example, since this is the National Cancer Institute, a 64-year-old with GE reflux, gastroesophageal reflux, and depression begins chemotherapy, etoposide and cisplatin for squamous cell carcinoma of the lung. And she also notes that she uses herbal supplements. She develops a severe drop in her platelet count that requires a platelet transfusion. A genome scan revealed no functional variants in phase 1 or phase 2 enzymes that would affect etoposide metabolism. Turns out that she was taking echinacea. She was also taking over-the-counter Tagamet for her GE reflux, and she was on Paxil for her depression, all three of which can interfere with P450 enzymes that metabolize etoposide, such as 3A41. So this is another example of how a genome scan wouldn't have found this particular type of personal medicine or precision medicine, but an exposomic scan could have found it. So how do we measure the exposome? Well, there's the internal exposome that I talked about, and we can measure nutritional factors, we can measure chemicals, metabolites, hormones, in the blood or urine. These are very traditional assays in many cases. Those are the targeted assays. And then there's untargeted assays, which are a little more unconventional, and I'll talk about a little more in a bit. But those are semi-quantitative, whereas targeted are quantitative assays. Those are what we are used to ordering, or the targeted assays. In our lab at Mount Sinai, actually, we've been really focused on trying to develop novel tests using novel matrices. So there's information in certain types of tissues, such as hair or teeth, that you can't get out of blood or urine. So we're trying to look at other ways to measure environment and actually maybe more informative. And Dr. Manish Arora, who's one of our faculty, who's a dentist, actually developed a method for going back in time using baby teeth to measure environmental exposures that occurred in utero. And hair has some similar growth properties as well. You can actually time past exposures using hair. And then there's the external exposome. And we tend to define that as anything that you can't measure in an assay. So these include things like air pollution, climate, weather, stress, light, noise. There are ways to do this using satellite remote sensing and mining of other public databases, including social factors. Technically, they're not physically inside the body, but they're certainly experience and cause a biological response. Oftentimes, because these are modeled, you can also go back in time, because when you do an air pollution model, you can go back to whenever those monitors were set up for that air pollution modeling, or whenever the satellites, if you're using satellite remote sensing, was set up for if you're using that type of model. But this is another example of a way to measure the exposome that's not necessarily an assay. And then there's the untargeted assay, so what we often call the internal exposome. This is typically done in blood or urine, plasma or serum, more specifically. I think it's going to be very important for diagnostics, and this is an example chromatogram. You can measure thousands of chemicals inside a specimen of blood or urine. There may be signature patterns that are specific to diseases. So instead of looking at a single chemical or a small group of chemicals, we can scan thousands to hundreds of thousands to one day millions. It's estimated that there's about 1 million chemicals inside each of our bodies. It should be kept in mind, however, that the 1 million that's inside my body isn't the exact same as the 1 million inside your body. There's definitely overlap, but there's also chemical signatures that are probably unique to the two of us. And this is an example untargeted chromatogram. There are also advantages potentially to looking at this both in blood and urine. Blood is more hydrophilic, so therefore, or excuse me, hydrophobic. So therefore, you're going to capture more of the chemicals that are less water soluble, whereas urine, the whole purpose of metabolism is to make a chemical more water soluble. So you tend to pick up the water soluble chemicals in urine. So there may actually be value to actually doing both matrices. Then in terms of the external exosome, there's lots of different things that we can measure using satellite remote sensing or mining of public databases. As I mentioned, this include things like weather, exposure to green space, so they can be positive environment. It can be air pollution. It can be traffic patterns and noise. It can be access to healthy foods using Google Maps. And then one of my favorite potentials for measuring the external exposome is actually social media. So if you know where someone is, you could actually look at some of the social media content, obviously with informed consent, to try to understand their social environment. This was actually a study one of our faculty did looking at mean tweets around the Boston area and geospatially plotting them. And then there's everyone's favorite, which is wearable devices and the Internet of Things. These are phone apps, cars, appliances, and these devices exchange data through the internet or with other connected devices. They are GPS detectors, so we know exactly where you are. Apple or Google know exactly where you are right now. They can even tell whether you're indoors or outdoors. But if we know where you are and we link that with the maps that we talked about earlier about air pollution, temperature, we have a more precise measurement of the environmental exposure to that external exposome. So how might we operationalize all this information, particularly for clinical medicine or precision medicine? Well, I think one day we'll go to the doctor's office and you'll give them your wearable device, whether it's a watch or a phone, and they'll download all the information into the electronic medical record. It'll have information on where you were, the GPS, coordinates, air pollution, temperature, physiologic data based on maps that are stored inside a supercomputer. And so we'll get estimates of your exposures to the external environment. It'll update your residential address if it's changed and how that affects the external exposome estimates. It'll note past infections for your medical record, what medications you're on, what now, what you've been on in the past, your history of surgeries, etc. If your occupational history has changed, you'll be updated and past exposures will be estimated based on your occupational history. And then you'll probably give a blood and or urine sample, and there'll be an untargeted screen to get a sense of your internal exposome, which will be linked to the genome scan that's already in your medical record. And then in the background, there'll be a supercomputer that does artificial intelligence analysis and generates risk scores from the exposomic data, and also looks at genome by exposome analysis for interactions that may actually predict health effects or response to treatment if you're there for a particular disease. So I think this is the future of precision medicine. So how do we get there? Well, right now in the environmental health world, most of our research is about prevention, and that's very, very important. And I certainly don't want to take away from prevention, but I think there's a whole untapped reservoir of research that could be done on the clinical area. And so in this graph, I'm actually separating the difference between public health and prevention research with clinical research. So if a disease onset occurs on the x-axis here, public health research tends to be in this space to the left of it, trying to prevent that disease from occurring. But from a physician perspective, we care more about the patient who already has the disease. At that point, prevention doesn't matter in terms of what we're going to do for that patient. So we need to understand how environment impacts somebody who has a disease, not whether or not the environment caused the disease, but how it's affecting the disease itself, which is a subtle distinction, but actually is something that we've done very little of. There's been some work in this area, but not very much. And this is the space, I think, if we're going to do precision medicine and exposomics, that we have to start to move into. So we have to start looking at the effect of the environment after a disease starts, and then move forward in time and assess its progression. So here's some quick and dirty examples. So maybe not dirty, but quick examples. So maybe we should start looking at environmental obesogen exposures as a modifier of glucose control in patients who have diabetes. That's different than saying that phthalates, which is an obesogen, is a cause of diabetes. We're saying that there are millions of people with type 2 diabetes in the United States. They're exposed to phthalates, whether it's from perfumes or from food products. And what are those phthalates doing to their glucose control and their insulin dose? That's an open question. There's very little to no research on that. And yet it's a very important question, because maybe if we modify their phthalate exposure, it'll be easier to regulate their glucose. As another example, maybe we should start measuring neurotoxic chemicals in patients with Alzheimer's disease, such as lead or mercury or arsenic. Maybe those chemicals are modifying the progression of Alzheimer's disease, and we can prevent that exposure if that's the case. There's almost no literature on this. There's literature on whether or not these chemicals are predictors of Alzheimer's disease, but how they affect Alzheimer's disease progression, there's almost no literature on. And maybe we can start looking at these untargeted chemical assays and look at whether or not we can predict who's going to have a side effect for patients with cancer who are on chemotherapy. Just like there's some really interesting work being done with light waves, and it turns out certain types of wavelengths of light actually reduce nausea and vomiting on patients with chemotherapy. There may be untargeted assays that have signatures that can help us predict who's going to have more side effects, or even who's going to have a better response to treatment. At the same time, we could start to use untargeted assays to detect diseases. I think eating disorders may be one of the earliest uses of this. So maybe we could detect somebody who has bulimia early on if a patient has weight loss or metabolic disturbances. In fact, I would argue that the use of environmental epidemiology in clinical studies is so wide open that most of the ideas are low-hanging fruit. Most of the studies have been done as risk factors to get a disease. Very few of the studies have been done to see what happens when you have an environmental exposure in people who already have that disease and how it affects the disease itself. So I think in the future, exposomics and precision medicine is going to enable better diagnostic tests that are more precise. It's going to help us understand the relationship between a patient's chemical, nutritional, physical, and social environment and disease prognosis. And it's going to do what precision medicine's goal is. It's going to help us optimize treatment based on one's exposome and genome so that we can maximize the treatment success, reduce side effects, and improve compliance. So what investments are needed? Well, I think most of the investment that's needed is actually in informatics. We need to get web-based data. We need the equivalent of OMIM, online Mendelian inheritance in man. We need to link together what are currently disparate public databases like PubChem and ToxCast and Tox21 and comparative toxicogenomic database, and even genomic databases like dbGaP, so that we can collate information, allow researchers to quickly search on information on their findings. A lot of the untargeted assays are going to give us information on what are frequently referred to as unknowns. We know something about them. We know their weight, we know their charge. There's probably 7 or 8 different variations on their chemical formula that give the same weight and charge, but that is information that we can use, and we can start to collate that information and also start to annotate which of these features from these chromatograms are associated with health outcomes. And we need a similar database in its organization for the external exposome as well. Instead of thinking of weather and light at night and noise separately, we need to integrate them into a single source that researchers can use. And we also need a reference database, very similar to the HapMap project. So because we have to understand that geography matters. The exposome in New York City is going to be very different than the exposome for someone who lives in Wyoming. Life stage matters. So children will have a very different exposure pattern than adults. And it's also time varying. Somebody who's 6 years old in 1999 will have a different exposome profile from somebody in 2020 because our environment changes over time. And we need to understand this exposomic reference data for both the internal and external exposome. And once we do that, I would argue that exposomics and precision medicine programs will look more like this, where instead of the current one-size-fits-all, we will stratify people on multiple characteristics, including their genome, but also their exposome and their behaviors and their geography and their life stage so that we can get more precise precision medicine. And that's my last slide.
Terrific. Thank you, Bob. That was very interesting. I have a few questions afterwards, maybe if others don't, but it'll be interesting to have some conversation about some of the things you raised in your talk. So I'd like to move on and introduce our next speaker, Dr. Jessie Buckley. Dr. Buckley is an associate professor in the Department of Environmental Health and Engineering with a joint appointment in the Department of Epidemiology at Johns Hopkins University. Her research focuses on environmental and molecular epidemiology investigations of the effects of early life environmental chemical exposures, including endocrine disruptors, obesogens, and complex mixtures on child health. She's a co-investigator for the Environmental Influences on Child Health Outcomes, the ECHO program, and she's a recipient of the NIEHS Outstanding New Environmental Scientist Award. Dr. Buckley, it's all yours.
Great, thanks. Well, I really enjoyed Bob's talk. I think that while my talk is more focused on environmental epidemiology in general, we both have this, I think, similar theme that kind of the future and things that we need to address are in a way tied to asking different kinds of questions. So when I was asked to give this talk, I started thinking about, what were the key gaps, a long time ago when we had John Snow, what were the kinds of things that he was working with, and, the simplicity of that work and the sort of data that was necessary to address this problem of the cholera outbreak in London was so simple and so beautiful when you think about the kinds of data that we're working with today. These are just some figures from some of my favorite recent papers. And looking at this kind of data versus a map with locations of cases shows how much harder it is to use our data in a way that can inform public health policy. It's become so massive, and we have had so many advancements in measurement and statistical analysis that in some ways it gets a little bit harder and further from action, which I think Bob also alluded to. So while our data are so much more complex, the kinds of questions that we're trying to ask and our goal is the same. We want to improve public health through interventions on environmental exposures. So the legend is that Jon Snow removed the pump handle, but the kinds of questions that we're asking don't lend themselves quite so easily to interventions. So how can we as a field continue to really directly influence practices and policies to improve environmental health? I think traditionally a lot of our work has been sort of problem oriented, and we've been thinking about what were the environmental exposures that have contributed to disease. And it kind of is a little bit focused on the past and very much focused on what is toxic and what are the hazards. Whereas the opportunity that we have is to start thinking more about solutions and how can the information that we've gathered and the studies that we do inform changes that could be made to reduce future disease. So this kind of switch from a problem oriented orientation to solution orientation seems like a subtle difference. I think that there's a really important implication. So this subtle shift has implications for the way we phrase our research questions, the kinds of hypotheses we generate, the design of our studies, and importantly, making our research results more relevant to policy and practice. So I include this as sort of, it's sort of a comical way of thinking about the kinds of papers that we write. This was posted by John Levy on Twitter last month, and I got a kick out of it. You can see the kinds of environmental health papers that he kind of alluded to, that we're doing studies on don't live next to the highway. Being exposed to a lot of things is worse than being exposed to one thing. The new chemical is worse than the old one. We found something in everyone's blood. Air pollution affects this health outcome you hadn't even thought about. And we found another method you can't pronounce to look at chemical mixtures. So, these are sort of comical headlines for work that we do that's actually really important. But I think the gap that we need to fill is showing that this important work is actually going to make an impact. And so for this, I think there's a few different kinds of ways that we can make our questions in environmental epidemiology more solution-oriented. So the first is to really start to think about how we can tie our exposures back to their sources. So if we're going to be looking at chemical X and we don't know where chemical X comes from, then we don't know how to intervene and improve public health. So even though we kind of think about this as an exposure science question, it's really critical for environmental epidemiology to understand how the things that we're studying relate to modifiable sources. And critical to a solution-oriented kind of framework is thinking about interventions. So intervention studies are also, we all know that they're very difficult in environmental epi due to reasons of ethics and feasibility and cost, but there are ways that we can emulate hypothetical interventions, and that's what most of my talk is going to focus on. Many of us are working on understanding modifiers, and I think, doing this even more with thinking about how we can assess other factors as potentially ameliorating the effects of toxic exposures is another way to be really more actionable and solution-oriented in our research. Additionally, thinking about vulnerable populations. So if we were to do an intervention on this exposure, who should we target that intervention to? So here again is sort of a framework of the traditional kind of approach that most of us are applying in our work, where we're assessing exposure to some environmental agent, and we're seeing if it is related to a health outcome. The solution-oriented approach would take into consideration how you might intervene on that exposure and how the intervention that is upstream of exposure affects the health outcome. So as I mentioned, this could be done ideally with a randomized control trial, though that's very difficult for many reasons. I think environmental epidemiology has been doing a pretty good job of trying to leverage natural experiments and quasi-experimental studies. For example, studies that have looked at the 2008 Beijing Olympics and the reduction that was in air pollution that occurred because of the Olympics and how that related to things like birth outcomes and asthma. So that would be a nice, a natural experiment. But those opportunities don't come along every day. So what is another way that we can estimate effects of interventions? And there have been major advances in epidemiology with respect to using G methods to estimate these effects. And I think that these kinds of studies have been adopted with much more greater frequency in other fields of epidemiology and really have a lot of promise for how we can be more solution-oriented in our environmental epidemiology studies. When we're talking about estimating effects of interventions, there's many different types of interventions that we could potentially look at. One would be, what is the expected change in some outcome if we eliminated the exposure of interest? So if we completely banned it. So there's a distribution of exposure in the population and that exposure is removed. That would be the ideal, but it's often very unrealistic. We could also imagine an intervention where we could shift exposures. Maybe exposure would go down by half or some other proportional factor for all individuals. But more realistic would be potentially implementing a exposure limit. In this case, we would say there's some sort of guidance value and exposure would be somehow limited to that value. And you can see that the type of intervention that you consider has a real difference in how it might relate to health outcomes, because in the case of a ban, you're removing exposure. In the case of a shift, everyone's proportionally going down. So even if you already had low exposure, you would remain having low exposure. Whereas if you implemented some sort of guidance value or a limit, you're really targeting those people with the highest exposures. So there aren't that many examples of using these sort of G-computation methods in environmental epidemiology, but this is a nice one that was published by Erica Garcia in 2019. And what this study tried to do was to use G-computation to estimate the incidence of asthma in children that would have occurred because of nitrogen dioxide and particulate matter that was observed in their study population, and then they compared it to what would have happened if levels of nitrogen dioxide and PM 2.5 were at levels prior to the major reduction in air pollution in Southern California. So this observed box is asthma incidence rate in their population. And if you look to the left in either panel, you can see what they estimated the asthma incidence rate would be had there not been reductions in air pollution in Southern California after 1993. So you can see there's a significant decrease in asthma incidence rates. Then if you go to the right, this is where they estimated effects of potential interventions on nitrogen dioxide and PM 2.5. They compared the asthma incidence rate under a hypothetical intervention that would reduce exposures by 10, 20, or 30%, or that would implement exposure standards, kind of looking back at the different kinds of interventions that I showed a moment ago. So this shows that while there's been a major reduction in the asthma incidence rate compared to what exposure levels used to be, there could be important future reductions if there were additional policies that were implemented to reduce air pollution in the future. So that is a nice example of this group of investigators trying to inform future policies by showing how much more you could reduce this burden of childhood asthma if you took it a step further and implemented an intervention on air pollution constituents. So recently I've been thinking a lot about this because I do a lot of work on exposure mixtures. And when we think about exposure mixtures, it's abundantly apparent that a particular source will give rise to many exposures. And so in thinking about mixtures in a solution-oriented framework, it really does make sense to think about how might we reduce exposure to the mixture by intervening on a source. So using kind of complex methods for exposure mixtures in concert with G-computation algorithms, we can start to think about how intervening on the source of the mixture might have an impact on health outcomes. And I'm just going to give a couple of very brief examples of ways that this might be conceptualized. So the first example is a project that I collaborated with Alex, Kyle, and Amy called Brenner, and was published earlier this year. And what we wanted to do was kind of demonstrate this approach of how you might be able to combine exposure mixture methods with G-computation to estimate the effects of an intervention. So we kind of came up with a hypothetical intervention that would reduce gestational metals exposure by decommissioning coal-fired power plants in Milwaukee County, Wisconsin. And so what we did was we linked data from EPA's National Air Toxics Assessment Model with birth records in Wisconsin. And then we looked at 6 particular metals that were measured in the air toxics and modeled how they were related to the emissions from coal-fired power plants. Then we used G-computation along with Bayesian model averaging to estimate effects on birth weight of decommissioning the coal power plants. So we had to have information on the air levels of the metals and also how they relate to the power plant in order to kind of estimate what an intervention on metals would do if you were to close the power plant. And then we estimated the difference in birth weight if you were to decommission the power plant versus not. And here you can see the mean birth weight with no intervention, or the observed birth weight in the population, was 3,191 grams. If you decommission the power plant, we estimated a mean birth weight of 32,559, and then the mean difference was 68 grams, and it was a significantly greater birth weight if we were to decommission the power plant. This work was kind of the beginning of kind of applying these sorts of methods in a proof of concept. Certainly there's a lot more work to be done in understanding how there could be other kinds of side effects of decommissioning these power plants, in particular if there were economic implications. So this isn't the final estimate, but it shows that you can take your data, you can do the traditional sort of mixtures methods and understand the dose response relationships, but then you can add on to that and take it a step further to see how an actionable public health intervention could affect health outcome under study. Another example in collaboration with Alex is to reduce gestational metals using a different approach. And in this case, we're conceptualizing how we could reduce the metals by implementing water filtration systems in an area of North Carolina where people are using well water that's contaminated with multiple metals. So in this case, we're linking data from the National Birth Defects Prevention Study to well water metals database, and then again combining G-computation with different Bayesian mixtures approaches and comparing the estimated prevalence of birth defects under different water filtration systems. And we can also evaluate cost effectiveness, which is of greater interest to policymakers oftentimes. So here we have identified several different kinds of water filtration systems that you could actually use in practice to reduce exposure to metals among pregnant women. And you can see that different purifiers have different efficacies for reducing different metals. So using one is not going to have the same effect as using a different one. So for this study, we're going to estimate the difference in birth defects prevalence if you were in the population compared to if you were able to reduce metals exposures with each of these different kinds of filtrations. So again, this is just an example of we're going to do all the regular work that we normally do in understanding mixtures and how they relate to birth defects, but then we're taking this extra step of trying to go back and see, well, how can we reduce those metals associated health effects with some sort of intervention that policymakers might actually consider. And by adding on the cost effectiveness, we can also show that putting a relatively cheap air filter or water filter on a tap could be very cost effective compared to the number of the burden from the birth defect cases. So those are examples of different kinds of interventions on exposures that you could conceptualize. Certainly there's other kinds of solution-oriented questions you could ask with respect to modifiers. So if we were to, instead of intervening on the exposure, the environmental exposure, we could mitigate those effects by intervening on, say, a beneficial factor like micronutrients or social support. We could also conceptualize interventions where we intervene on both. So what if you could decrease exposure and increase this mitigating factor? What kind of impact would that have on public health? I think a lot of us have been interested in critical periods of susceptibility, and you can also kind of use these approaches to understand when you should intervene. Are there heightened periods of susceptibility? What is the benefit of intervening, say, during pregnancy versus early childhood or at other time points? I think a really critically important question that you can answer with these kinds of approaches is understanding what the expected change in the outcome would be if you targeted an intervention to a specific population subgroup. And I think a lot of times when we think about effect modification, we'll find that associations between some exposure and some outcome are not modified by something like socioeconomic status or race, ethnicity, and then you stop there. But if a certain subgroup is differentially burdened by our environmental exposure, then they'll still have the greatest impact. And using these sorts of methods really highlight that if we want to address environmental justice issues and health inequity, we need to target interventions to those that will benefit the most. So there are, as with any method, there's some limitations or challenges. When you're estimating effects of hypothetical interventions with these G methods, you really do need good information on how the intervention affects the exposure. And I think, as with any environmental epidemiology, exposure assessment is the Achilles heel. And in this case as well, we've had to make some simplifying assumptions in our work, but as we focus more and get more data on how exposures and sources link, it'll be easier to do that. It's important to consider unintended effects of the intervention. So it's easy to estimate the effect in any study of some exposure and some outcome and control maybe for a co-exposure. But in this case, it's almost an opportunity that you can really try to consider the unintended effects of an intervention. So if you're going to institute a congestion charge that might cause less traffic, you also might have an unintended side effect of more physical activity. So that could be two beneficial exposures that would result from an intervention, not just the effect of reduced air pollution. These methods can be computationally intensive, but the software is becoming increasingly available. There's definitely code and macros available now. And it requires assumptions, basically the same assumptions as any other method, but there's a few additional because of the additional steps. I think it has major advantages in terms of informing public health action, being able to leverage, we still have these important questions about mechanisms. We have important questions and large data sets, but we can use those and then kind of add this on as an extra step that provides a result that is easier for stakeholders to understand and that can potentially have more of an impact in terms of policy. And we can even contrast in different kinds of interventions to figure out how to spend our limited public health resources for the greatest impact. And importantly, I think in this framework, it's really straightforward and important that we can start looking at the sources of exposures and also the things that contribute to environmental health inequities and be able to estimate the effects of interventions on those, which give rise to the inequities we see in exposure, but in a way that we can look at it more formally. So I guess I'll just close with kind of a challenge that we consider adopting a solution-oriented framework in our work. Even if you don't estimate effects of potential interventions, you can take the time to think about how you might frame a research question, and you can make small changes to how you approach your work. Like, for example, I recently did a study looking at ultra-processed foods as a source of phthalate exposures. So instead of just looking at consumption of ultra-processed foods, in a solution-oriented way, we also looked at what happens if you consume minimally processed foods. And that's sort of like just another way of framing the question in a way that's a little bit more positive and a little more health promoting. And I think it's important with these, thinking about this is to collaborate with exposure scientists and engineers that are working on these kinds of interventions. And that way we can partner to understand what information we need in order to estimate these effects, and then potentially help them determine which are cost-effective solutions. And then, of course, as with any real study, we want to engage with communities and stakeholders to ask important questions that matter to them. Because in the end, if we're going to be informing interventions, we're going to be helping real people. And so we want to be able to address the questions that are of importance. And so that's where I will close, and feel free to reach out if you have any questions.
Terrific. Thank you very much. Appreciate the time everyone has spent, both our speakers and our attendees. I see we do have a few questions that I could ask to our speakers, and obviously if any more questions come in, please feel free to ask them. First question for Bob was, could you talk about the methodological challenges to assess cancer risk as an example, if we are to leverage the technologies from an exposomic perspective? I assume maybe talking about some of the laboratory techniques, maybe as a reference to.
That's a great question. Yeah, to do that kind of causation research, the complicating issue is the latency between exposure and the onset of the disease, but not only the onset, but also the diagnosis of the disease, because it's probably itself is latent for a while. Some of the work that Manish Arora has been doing with teeth may help to address some of that. So it turns out teeth grow like trees, and there are growth rings in your teeth that start around the second trimester of your life, and they're formed once a day, and chemicals get deposited in them. And because of the calcium appetite matrix, they get trapped. And so it is possible, theoretically, if you have deciduous teeth, to reconstruct exposure in the during pregnancy and up until the point at which the tooth was expelled, which will be somewhere between 5 and 11 years of age. And so if we think the latencies for a lot of important cancers are from early life exposures, creating biobanks, partnering with physicians or families and communities to just collect teeth and store them so that we can do nested case control studies, not unlike the way that a lot of states do, dry blood spot analysis on relatively newborn babies. That might be one way around it. And also the dry blood spots might be another way around trying to get to those early life exposures, at least the chemical exposures to carcinogens, so that we can start to understand what role these chemicals play and overcome that barrier of not being able to go back in time years to when that exposure happened.
Great. I have just a follow-up on that. And you mentioned using teeth or hair as a way to assess sort of get a sort of longitudinal measure of exposures. And I'm just wondering, what are some of the limitations of that matrix in terms of the types of exposures you can measure and how well you can pinpoint timeframes?
Are you talking about teeth or hair or both?
Either.
Well, teeth, it's been very well worked out with respect to trace elements, which do tend to accumulate in teeth. It's something Dr. Rohrer is working on with organic chemicals, but we do know organic chemicals do deposit in teeth because tetracycline and other types of antibiotics actually do get stored into teeth. I'm sorry, my fire alarm is going off, but the house is not on fire. And with respect to hair grows at about 1 centimeter a month. So it's longer term than blood or urine, but it's not going to go back years. It's only going to go back a few months. But I think that's potentially useful for, say, pregnancy studies, where if you collect hair at the time of delivery in the mother, and if it's long enough, you may be able to get estimates of exposure throughout pregnancy. Now, there are concerns about contamination. I have mixed feelings about that. We use a lot of these silicone wristbands in which we allow chemicals to just fall on top of the wristband, and we call that exposure. When we use hair, we call that contamination. I'm not so sure that we actually shouldn't wash hair before we analyze it, because technically you were exposed to whatever was on that hair. But those are some of the limitations to hair and teeth.
Okay, thank you. Another question here. Jessie, could you just give like a very high level explanation of the G computation method?
Sure, yeah. Basically what you're doing is you're estimating, it's sort of like standardization, where you're estimating the probability of the exposure, outcome, and covariates, and censoring if you have censoring, and it's a survival model. And then you use those models to create a simulated data set in which you can intervene on the exposure by changing the exposure level, and then you predict the outcome based on those estimated conditional probabilities. So there's the underlying models that you use to predict exposures and outcomes and covariates can be very complex if you want them to be. So that's one reason why we like to use, for the mixtures approach, we can use really complex methods. You could use a BKMR or some other complex method, and then use that to estimate the effects of the intervention within a simulated data set where you've intervened on the exposures. It's hard for me to really, I need to practice explaining it in simple terminology, but you can just imagine that you're using the model to predict the outcome under different exposure concentrations.
And then along those lines, another question, it says, how do you include different chemicals in the G methods to be sure to specify the correct model? Do you first look at the relationship between each chemical with the outcome and interactions between co-exposures?
Right. So if you had a single exposure, you could use like linear regression model. But in the case where we had multiple exposures, we used Bayesian model averaging so that we could create a kind of a joint exposure response surface, but still be parsimonious and not include terms that were not necessary to predict the exposure response relationship. You could use really almost any model with as much complexity as you want, to the degree that your data are large enough and you can support the terms.
I'm going to open this question to both of you. Question here is have you thought about interventions that might mitigate the harm of environmental exposure, since exposure reduction methods may be difficult for groups in disadvantaged neighborhoods or limited access to remediation methods? Things such as maybe like dietary modifiers or behavioral interventions.
That's a great question. I think one of the things I think we don't quite understand yet is the degree to which systemic racism has created disparities through segregation and redlining. I think we only know the tip of the iceberg, so to speak, and I think understanding through exposomic methods the differences between communities is probably one of the first steps. And then we can start to look at the sources, and we'll have to mitigate those sources. But I think right now we don't really understand just how big the problem is. And so I think there is a lot of work that has to be done just in measuring these environmental factors.
Yeah, I agree completely. And I think if there are some cases where we have some idea that there's a modifying factor where we might not be able to intervene on the exposure, but for example, you could provide folate during pregnancy. And that is an example that I was thinking about, where you could estimate the effect of an intervention on a nutritional exposure that would mitigate the effects of the environmental exposure that might be harder to intervene on. But certainly going more upstream and thinking about other kinds of interventions is also a place where, with more data, we can do a lot more work.
This question was directed at Bob, but I'll leave it open for you too, Jessie, if you have any comments. The question was regarding interactions between the genome and exposome, and asking, what do you think the best way to study this interaction is?
Oh, that's a great question. I think the interesting questions about genome by exposome interactions is, we didn't talk about critical windows, but there's clearly life stages at which exposures are more toxic than at other life stages. So the best way to do genome by exposome interactions, I would argue, is you're going to have multiple discovery sets with exposures at different life stages and multiple replication sets as well, because I think the gene environment interactions are going to differ based on not just the exposure, but also the age at exposure. And I think there hasn't been a lot of methodological work in that area, but I think it is a really interesting area because it's not going to be straightforward. It is clearly going to vary by life stage and probably other factors that we haven't yet discovered.
And one more question here for Jessie. In your description, you describe some of the assumptions that go into the models, and there was a question about how do you incorporate uncertainty, particularly in the exposure assessment, into the G method models that you were using?
So you can incorporate uncertainty because the way that the method is run, you sample and run like multiple iterations. It's kind of the Bayesian part, of course. You can incorporate uncertainty through the Markov chain Monte Carlo, and then with the G computation part, you can run multiple iterations and include the uncertainty in the exposure assessment is incorporated through the bootstrapping. But I think that there could be more formal ways of incorporating uncertainty. And I think when I was thinking about this talk, and one of the directions I thought about going in was around measurement error, because clearly that is still a major problem for environmental epidemiology. And so I think it would be really, there are a couple of people I know of that I think are trying to more formally incorporate those, and kind of, especially with a mixtures framework, thinking about correlated exposure measurement error. So I think there's opportunities to more formally address that.
Great. And then just since we still have a minute or two here, maybe just one more question that was for Bob. It's going back to your warfarin example. It says, could you comment on the warfarin example, the results of clinical trials and the role of gene testing in clinical practice?
Well, I don't do genetics research, but my understanding is polygenic risk scores are proving to be much more predictive than individual genetic variants. I think by extension, polygenic or polygenome by exposome risk scores will probably be even more predictive than polygenic risk scores. It's because there's different ways to be a slow metabolizer of warfarin. One is genetics, but another is if maybe you drink a lot of grapefruit juice, or maybe you're taking a medication that inhibits the same P450 enzyme. You will be functionally similar to somebody who's a slow metabolizer, but the genomes won't pick it up. So I do think that the value of genome by exposome is we're more likely to actually be able to detect and predict when a problem is going to occur.
Right. Okay, so I see we're coming up on 2 o'clock here. So I just want to thank our speakers again. I think you guys have given us a lot to think about in terms of how we should be pursuing research in the environmental health field moving into the future. And as we mentioned before, we will at some point be posting this these talks again for viewing, so people can access them in our archives. And I just want to thank everybody and have a great afternoon.
Thank you.
Thanks.